All right, thank you, Lana, for that introduction. And thank you all for joining us today. Today I'll be talking about our ONCOREF molecular reference standards. So just as a quick outline of what we'll be discussing today, first I'd like to start with a little background on why we generated the ONCOREF series and also why we chose the particular pathway that we engineered with these products. At that point, I'll then talk a little bit about isogenic cell line generation using CRISPR Cas9 and how we employed that for the generation of our molecular reference materials. And then I'll talk about some of the applications of our molecular reference standards. And then at the end, because we can't address all of your needs in terms of molecular reference standards with our off-the-shelf products, I'll talk about some of the other custom services that we also offer at Applied Stem Cell. So the reason that we developed these series is that there's really a need for better diagnostic assays for cancer research. And so in 2016, in the US alone, it's estimated that there will be 1.7 million new cases of cancer diagnosed. And approximately 600,000 people will die from this disease. So this amounts to approximately 1 person per minute, or approximately 30 people over the course of this webinar. And the National Cancer Institute has estimated that approximately 3 to 35% of premature deaths that are due to cancer could be avoided with better screening and diagnostic methods. And this number could be even higher if we had better methods by which to treat these diseases as well. And so the goal for us in developing this series was really to develop a toolkit that can be used for enabling research into better diagnostics and treatments. And because of this, we developed the ONCOREF product series. So the ONCOREF product series consists of 51 different mutations within the MAPK pathway that we engineered using CRISPR Cas9. All of the mutations that we engineered are very prevalent mutations within cancer, and they were identified from the COSMIC database of somatic mutations. And so this features 32 mutations in EGFR, 12 mutations in KRAS, and 7 mutations in BRAF, and these have been engineered into fairly common colorectal cancer cell lines, RKO and HCT116. So because we have both the parental cell lines as well as the engineered cell lines, we can offer these as isogenic positive and negative controls. We've also engineered these cell lines to have homozygous mutations, so we've engineered the mutation into all copies within the cells. And the mutations have been introduced using footprint-free technologies, and I'll talk a little bit more about these later on. And we provide these products in a number of formats, including genomic DNA, a number of FFPE products, including slides and scrolls, as well as the cell lines that we use to generate the ONCOREF series. And so the pathway that we chose in starting off this series was the MAPK pathway, and there are a couple reasons for this. So if you look at this pathway in the literature, the MAPK pathway has been reported to be involved in a number of different cellular characteristics, including proliferation, resistance to cell death, differentiation, migration, and invasion. And these are all hallmark characteristics of cancers. And so it's not surprising that this pathway is frequently mutated in a number of different cancers. So if you look at pancreatic cancer, these cancers typically have KRAS mutations in greater than 90% of malignancies. There are BRAF mutations in approximately 50% of malignant melanomas. And then in non-small cell lung carcinoma, we typically see EGFR mutations in about 20% to 50% of cases, with the 20% being present in Western populations and approximately 15% in East Asian populations. And so we wanted to design a molecular toolkit that could be used to aid in the diagnosis and study of these mutations within the MAPK pathway. So to engineer these products, we used the CRISPR Cas9 platform. So Cas9 is an RNA-guided nuclease that uses a guide RNA, which is complementary to a region within the genome. The guide RNA directs Cas9 to the site in the genome, at which point Cas9 forms a double-stranded break. So at this point, Cas9 has pretty much done its job, and the cell chooses one of two pathways by which to repair the double-stranded break. So if no DNA repair template is available, the cell typically chooses the non-homologous end-joining pathway, in which it basically tries to stick together the two ends of the DNA that were cut. So this pathway is very error-prone and often results in indel formation. In contrast, if a repair template is available, the cell can actually use that template as a repair for a repair pathway through homology-directed repair. So this is actually a very precise way of modifying the genome or correcting the genome, and this is what we use in order to engineer our cell line series. And one of the benefits that I want to mention about Cas9 technology is that it allows for footprint-free genome engineering. So there are actually a number of approaches that are currently used for modifying the genome, and several of these techniques are used for generating molecular reference standards. So I just want to point out one example here, which is the recombinant AAV-based technology. So what this technology is based on is the genome of adeno-associated virus. So basically what this method uses is it uses a homology repair template that's based on the genome of adeno-associated virus that contains a left homology arm and a right homology arm to the region in the genome that you want to edit, along with the mutation that you want to introduce. So the drawback of this technique is that it's actually very inefficient. And so in order to bump up the efficiency of homologous recombination, you often have to add a selection marker in order to increase efficiency. So the problem with that is that even after engineering the cell line and removing the selection marker using Cre recombinase, you're still left with a genomic footprint. So you still have a single LOX-P site that's left over. So in contrast to this, with the Cas9 technology, because the technology is so efficient, we're able to introduce the mutation that you want to engineer without the presence of other selection marker footprints. And so this is actually really important if you want to develop assays for diagnostic applications. The reason being is that if you're trying to develop an assay to detect a mutation, you don't necessarily want to have to worry about how the presence of a genomic footprint may impact any primer or probe designs that you want to implement, or potentially your sequence base calling algorithms if you're doing next generation sequencing applications. And so we think that this is really one of the strong points of our ONCOREF series of cell lines. So the way that we engineer these cell lines is that we take the parental cell line population. We then do Cas9 transfection along with our single-strand oligo donor. At this point, we'll get a mixed population of cells, some which have the mutation and some which don't. At that point, we then move on to single-cell cloning. And in that process, we're able to identify cells that have the mutation that we're looking for. With those populations that we've identified, we then expand those and confirm them by Sanger sequencing to make sure that the mutations are correct. And then at the end, we have a cell population that we've genetically validated. It's clonally homogeneous because it's derived from a single cell clone, and also it has a homozygous modification for the mutation that you're interested in. And so we can then pair this with a parental cell line to generate an isogenic pair, either for assay development or for screening applications using the parental cell lines. And so one thing I want to point out is that because we're using the Cas9 platform, we're able to get homozygous mutation of the genetic modification that you want to introduce into the cell line. And so this can actually be very useful for assay development because it allows you to look at the specificity of your assay. So in many cases, you may be developing primers or probes that are designed for a particular mutation, but you may not know if they have cross-reactivity with other mutations within that gene. And so by being able to provide you with 100% mutant lines, you can really start to assess whether or not you're having off-target and cross-reactivity with your assay protocols. So just to show you a few examples of the different types of mutations that we've engineered using this technology. So in the first panel on the left, I'm showing you a point mutation in which we've engineered a G to T mutation within exon 20 of EGFR. In the middle panel, I'm showing you a precise insertion in which we've inserted an in-frame GGT mutation within exon 20 of EGFR. And then in the third panel, I'm showing you a case in which we've engineered a precise deletion, in-frame deletion of 18 nucleotides within EGFR. And so this just gives you an example of kind of the broad diversity and also the versatility of using the Cas9 platform for cell line engineering. And so we've engineered a number of different mutations, as I mentioned, using this platform. So we've engineered a total of 51 mutations in the MAPK pathway. So this features 32 mutations in EGFR, which includes 3 mutations in exon 18, 22 mutations in exon 19, 5 mutations in exon 20, and 2 mutations in exon 21. We also feature a number of mutations in KRAS, in which we focus on mutations in codons 12 and 13, which are particularly a hotspot for mutations in the KRAS gene. And we also have a number of different mutations in BRAF, focusing primarily on the 600 valine residue of the protein. And one thing I want to point out is that all the mutations that are starred in blue are mutations that are exclusive to applied stem cell. So these are reference standards that you can't find at any other company. And I particularly want to focus just briefly on our exon 19 deletion panel, which is very specific to applied stem cells. So within exon 19 of EGFR, we've engineered 22 deletion mutations, and these are all in-frame mutations that occur within the kinase domain of EGFR. And 21 of these mutations are specific to applied stem cell. And I just want to show you a couple more examples just to show you some examples of the complexity of mutations that we can engineer using this approach. So here, instead of just doing a single deletion, we've engineered a combination of 2 deletions within EGFR. So here you can see the sequence alignment data for this. And then the bottom one, we've combined a series of 2 deletions as well as a single nucleotide change within the gene. And here again, you can see the alignment information for this mutation as well. And these mutations within exon 19 actually have clinical significance as well. So it's been shown that deletions within exon 19 can actually result in enhanced sensitivity to anti-EGFR therapies. And so these mutation panels can be useful for a couple purposes. One is for biological applications, using the cell lines to better understand this pathway and why these mutations impart sensitivity. And the other is to use as potential reference materials for developing assays by which you can detect these deletion mutations within patients. And so far I've focused on the cell line engineering and how we validate these cell lines using Sanger sequencing. And so now I'd like to transition a little bit to talking about their application to reference materials for diagnostic assay development. So there are a number of reasons that you might want to employ reference materials in assay development, and I've listed a number of those here on the left-hand side. So some simple ones are simply for optimizing a number of protocols associated with assay development, whether that's identifying and optimizing your reagents, whether that's primers and probes, or library preparation procedures for next-generation sequencing. They can also be useful as materials for establishing limits of detection, as well as a number of quality control and assay reproducibility applications. And so one thing that I want to quickly mention here is that all of these reference standards that we're developing at Applied Stem Cell are for research use only. So there are a number of reference materials that people currently use for assay development, and I'd like to outline a few of those on this slide. In here I've categorized them by 2 characteristics, how reproducible these reference standards are, and then how relevant they are to biological specimens. So because there aren't a very large number of precision reference materials that are currently on the market, a lot of times researchers will have to resort to using materials like plasmids and amplicons for assay development. So the benefit of these is that they're highly reproducible, but the problem is that they're not very relevant to biological specimens, simply because plasmids and amplicons don't really look like any materials that you would find in a biological tissue. So now in contrast, other researchers are using tumor xenograft models for assay development. So these have the benefit of being representative of biological specimens, but they're not very highly reproducible. So oftentimes you'll get differences between the quality of the xenograft or also simply the morphology and characteristics of the xenograft across different xenografts that are generated. And so at Applied Stem Cell, we really wanted to focus on these bottom 2 categories, which are genomic DNA. Genomic DNA is, for obvious reasons, a very good biological specimen, simply because it's simply the genetic makeup that makes up our cellular genome. It's also highly reproducible. So in addition, we also offer FFPE blocks, and I'll talk a little bit more about these later, but these are are tissue blocks that are meant to simulate some of the biological tissues that you would encounter within the clinic. So first, I'd just like to start with some applications of genomic DNA. So genomic DNA is really a perfect starting point for assay development because it's high purity. It's also very high quality DNA. And so I would just like to outline one particular application that we worked with a researcher to develop. So we had a researcher who was interested in using genomic DNA for developing an assay, a next generation sequencing assay that they were doing. And so in this particular case, the researcher was trying to implement a molecular barcoding approach for reducing sequencing errors within next generation sequencing data. So the way this method works is shown schematically here. So initially you start with a population of DNA from your cell lines or from your clinical sample that has both real mutations, which are shown in gray dots, as well as a number of wild type alleles. So we then, in this process, the researcher then attached molecular barcodes to the end of the DNA, such that each copy of DNA gets its own unique molecular identifier. So at that point, you then amplify and sequence the DNA, and in this process, you have both the true mutations that are still present in gray, as well as a number of false mutations or sequencing errors that arise due to PCR errors or errors in the sequencing process itself. However, because you've barcoded these sequences, you can now group them according to the molecular barcode and eliminate, just by statistics, all of the false mutations while retaining the true mutations within the sample. So this particular researcher spiked in mutant DNA to approximately 2.2% within a wild-type DNA background. And here is just showing the raw sequencing data of the region that they were interested in. So here what you're looking at is you're looking at the observed mutation frequency as a function of base position within the gene. And in red, we've circled the mutation that was spiked in at approximately 0.2% within the sample. And so here what you can see is that if you just look at normal next generation sequencing data, the true mutation is actually buried within the background noise of the method. However, using the molecular barcoding approach, the researcher was very easily able to distinguish the true mutation from all of the false mutations within the sequencing background. And so this just illustrates one potential application of using some of these genomic DNA reference standards for assay development. So with that, I'd like to transition to talking a little bit about our FFPE reference standards. So just to give you a little bit of background on formalin-fixed, paraffin-embedded samples, this is a process that was really established by molecular pathologists in order to preserve biological specimens. And so essentially what you do is you fix the biological specimen in formalin, and then you embed it within a paraffin block. And by doing this, it really helps you to preserve tissue morphology, and you can see some examples of that in these images on the bottom, showing some applications in immunofluorescence staining, immunohistochemistry, and also in situ hybridization. And this also allows for the banking of tissues for analysis at later times. But the problem with this approach is that while it's very good at maintaining tissue morphology, if you want to use it to apply the samples to different types of analytical methods, such as nucleic acid detection using PCR or sequencing types of analyses, there are often a number of different challenges with these samples. So one of the challenges that often when you try to extract DNA from these samples, it results in low yields of nucleic acid. And the other problem is that DNA is often significantly degraded when you analyze these samples. And this is because the fixation process often results in double-stranded DNA breaks, which results in fragmentation of the DNA. In addition, it's well known that cytosine deamination can also result during the fixation process. So this results in the conversion of cytosine to uracil, which then, if you do sequencing, results in the occurrence of a C to T mutation when there wasn't actually that mutation within the original sample. And so if you're looking at and analyzing samples like this, it's really great to have a reference material that mimics the type of changes that you would see within these samples. And so in order to do this, we generated an FFPE cell line block product. And so the way that we generate this is that we take cultured cells, we then we then detach those and embed them within a mold that we then put inside of a paraffin block. From that block, we can then generate FFPE slides or FFPE scrolls that you can then use in your applications. And one thing I want to point out here is that if you're using currently FFPE tissue sections in some of your assay development, our cell line series offers significant benefits over that. One of the reasons is that if you're using these tissues that are embedded within paraffin blocks, oftentimes you'll get different section sizes as a function of depth within the block simply because of the tissue morphology. And so that can result in a lot of variations in terms of extracted DNA. Whereas with our block series, because we're using a cylindrical mold, you get very consistent results throughout the course of the block. And I'll show this a little bit on the next slide. So with our FFPE block series, as I mentioned, we're able to get very consistent results across blocks. And this is very beneficial if you're looking to try to do assay development. And so here I'm just showing 3 examples where we generated, using a single cell line, 3 different blocks of FFPE materials. And here, what you can see is you can see a low magnification image where we're showing the full cross-section of the block. So this is what the entire cylindrical section looks like. And then on the bottom, we're showing a magnification. And so from this, what you can see is we get very good density of the cells within the block, and we get also very good uniformity across the entire cross-section. So in addition to having very good reproducibility across blocks, we also have very good intra-block consistency. So here what I'm showing is we've taken one of our FFPE cell line blocks and we've cut it into 5-micron sections for staining. And here I'm showing 3 different slices from within that block. So typically from our FFPE blocks, we can get about 500 5-micron sections. So here what I'm showing is a section from near the top of the block, section 1, a section from near the middle of the block, section 200, and then also a section from near the end of the block, section 400. And so with these, you can see again that we get very good consistency of our cell packing density across the entire block, both in terms of the radial direction as well as throughout the cylinder. So in addition to our staining applications in slides, we also offer FFPE scrolls. So on the previous slides with the staining, what I was showing you is 5-micron sections that were mounted onto slides for staining applications. We also offer scrolls, and these are typically 15 to 20 micron sections that instead of mounting onto a slide are just allowed to curl into a cylinder, which is why they're called scrolls or curls by people who use these tissues for analysis. And so these are really an ideal source of material for optimizing your DNA extraction protocols, as well as your analytical protocols for looking at FFPE samples. And so here I've just showed you an example of some PCR data in which we were specifically looking at the BRAF V600E mutation by using a TaqMan qPCR assay. And again, I'm just showing you some consistency between our block generation. So here I'm showing you some 2 replicates from the first block that we generated using this cell line, as well as 2 replicates from the second block that we generated using the cell line. And from the qPCR data, you can see that we get an almost perfect overlap between these 2 blocks. So from our FFPE samples, in terms of DNA extractability, we're typically able to get around 600 nanograms of DNA using a double-stranded DNA-based quantification method. And one thing I want to point out here is that if you're using these types of samples for your analytical methods, you want to be very cognizant of the type of method that you're using to quantify the DNA in these samples. So in terms of the nanodrops are used fairly frequently in labs to quantify DNA. But the problem with this approach is that it's based on absorbance. So any sort of background material or impurity that's in your sample that has an absorbance in the UV wavelengths can interfere with quantification using that method. Whereas the QuantiFluor method is based on double-stranded DNA, and so it's very specific to DNA detection. And so we recommend using the QuantiFluor or some sort of DNA-specific analytical method for quantifying DNA from these samples. Okay. So in addition to our reference materials, we also offer a number of custom cell line generation services. So if we don't currently have mutations that are offered with our off-the-shelf products, we can also engineer them for you using the CRISPR Cas9 platform. And so the method to engineer these would be essentially the same process that we currently use for the generation of our current series of projects. So we start with Cas9 transfection. We then do single-cell cloning and genotyping to identify positive clones. And then we expand those and confirm them to give you the clonal population that contains the mutation that you're interested in. And so these are our full-service cell line engineering projects that start from design of the guide RNA constructs through genotyping, expansion, and positive clone identification. And the timeline for these projects is typically around 3 to 5 months. In addition, we offer a number of custom FFPE slide services. So the benefit of this is that you have the benefit of the consistency and reliability of our block generation, but you can apply it to your own cell lines, which you've been using within your own lab for assay development. And so using this, we offer both single and multi-spotting capabilities to meet your research needs. And so here I'm just showing a number of different examples of different cell lines that we've successfully made FFPE blocks from using our established protocols. And you can see that we've done this with a number of different cell lines and obtained similar cell densities and consistencies across blocks. So just to summarize what we've talked about today, I introduced you to some of the MAPK reference standards that we offer at Applied Stem Cell, which includes 51 different cell lines within the MAPK pathway. These cell lines are offered as isogenic positive and negative controls for both screening applications as cell lines, as well as reference materials for assay development. And they're available in a number of formats, including genomic DNA, as well as FFPE blocks, slides, and scrolls. The mutations are engineered as homozygous mutations, and they're also footprint-free, and these are really two key features of our MAPK and ONCOREF platform. And then we also offer a number of custom cell line FFPE services at Applied Stem Cell. And so with that, I'd like to thank you for your attention.